started on the lecture today. Does anybody have any questions regarding what we've learned so far? All right, awesome. So today we're going to learn about photosynthesis. All right, we're getting a little bit more complex into plant biology. So I have tried to break it down as simplistic as I can make it. But if you have a question along the way, just let me know because it's likely other students will have that question too. All right, so just as a review, all right, we're looking at photosynthesis. We're looking at a chloroplast. All right, so I threw this slide up here. I know we've already seen this slide just to kind of reorient ourselves to where we are looking. All right, so we're going to start talking specifically about these light reactions and the Kelvin cycle. So remember the light reactions, they are taking place in these thylakoid membranes, right? Whereas the Kelvin cycle is still taking place in the chloroplast, but it's taking place in the stroma. All right, so outside of the thylakoid membranes, but still inside of the chloroplast. All right, we're going to start with the light reactions, all right, because it's typically the first thing that the plant will do. All right, we're using light energy and water, right, to create oxygen, NADPH, and ATP, which we will define a little bit more clearly here. All right, so I did throw up the molecule here of NADPH and ATP. Basically, I already said everything that was on the slide here. It's kind of a redundant slide. All right, but what is NADPH? All right, so NADPH is a molecule that can carry electrons. All right, so remember when I said we had light coming in and it's exciting those electrons and it's moving it to a larger orbital around the nucleus of an atom, and sometimes those electrons can be captured by other molecules. All right, so this is one of those molecules that can capture electrons and move them around within a cell and donate them to where they are needed. All right, so essentially this molecule is a molecule that can provide energy for cellular reactions. All right, so a lot of times we're going from this form of NADP+, we are adding on a hydrogen molecule and some electrons, and that's what turns it into NADPH. All right, so we'll see this towards the end of the light reactions. All right, I found this amazing meme the other day that kind of explained NAD+, which is found in cellular respiration, but it's essentially the same exact molecule as NADPH, all right, or NADP+, does the same thing. It can carry the electrons for you. All right, ATP should be a molecule that you are more or less familiar with. You've probably at least heard the term before. All right, so ATP is kind of the energy currency of the cell. All right, it can store and transport a lot of energy due to the chemical bond in one of its phosphate groups within the molecule. All right, so we're not going to dive into any more deeper than that. Right, but know that it can carry energy and provide energy for cellular processes within the cell, many different cellular processes. All right, I just listed a couple of them here: cellular signaling, synthesis of different chemicals that the plants might need. Maybe it's synthesis of hormones. All right, and we're using that energy that's found in the bonds within this molecule. All right. So now that we've gone over NADPH and ATP, let's look at the light reactions as a whole. All right, yes? Are you recording this? I am recording it, yes. Thank you for reminding me. I actually remembered today. Thank you. All right, so light reactions as a whole, we have some major molecules that are present, and we will go through this step by step. All right, but I kind of just wanted to give you an overview. All right, so first of all, we are using water. Right, H2O, right, in our little diagram here, we can see it at the very bottom of this photosystem 2. All right, we have a couple of different photosystems that we will be looking at, right? One is photosystem 1, and the other is photosystem 2. All right, I often abbreviate them as PS1 and PS2. So if you see that abbreviation, know that I'm talking about photosystems. And they are essentially a molecular complex, right, that's embedded within these thylakoid membranes. Another complex that's fairly important in the light reactions, right, is the cytochrome complex, which is in blue in our image up here. Right, that is another molecular complex, right? So it's a very large molecule embedded within the thylakoid membrane. We have an enzyme called NADP plus reductase, right? We're going to find that towards the end of the light reactions. And then we have another very large enzyme, all right, that is embedded within the thylakoid membrane called ATP synthase. All right, and this is an enzyme that's going to help us create ATP. All right, so when we're looking at the light reactions, we'll be looking at all those molecules, but I want you to focus on the pathway that an electron is going to take. All right, so we have this excited electron. It has been captured by molecules, and then it's going to go through the light reactions. So as we're going through, I want you to focus on that electron up here. All right, you can kind of see that the pathway that it is taking, it is going to bounce from essentially molecule to molecule to molecule, and we will follow it along its path. All right. So kind of the biggest and most important molecular complex are these photosystems. Yes?
Oh, I mean, maybe you'll explain this, but why does it go through photosystem 2 first?
Yes. So on our slide here, photosystem 2 is the first step in photosynthesis, right? But it's named 2 because it was discovered second. Right? Photosystem 1 was discovered first, which is why it has 1, even though it does not happen first. Right? So remember that when you're thinking about these photosystems. Right? So we have 2 of them. Photosystem 1 discovered first, but occurs second. Right? Photosystem 2 discovered second, but occurs first. Right? So just to make it a little bit more confusing for everybody. All right, so we have these photosystems. They are embedded within our thylakoid membranes, and it's at these molecular complexes where that light is going to come in and excite the molecules, all right, just like we talked about yesterday. So if we look a little bit more closely at a photosystem, right, it's going to have 2 main components. It's going to have an antenna complex. Right, so if you remember yesterday we talked about antenna complexes. That's where there's a grouping of pigment molecules that are grouped together where the light comes in and can bounce between the different pigments. Right, we can see that here. We have light coming in. These round circles represent chlorophyll molecules. And that excited electron, it's bouncing between different pigment molecules. All right, that would be the antenna complex part. The other part is the reaction center. All right, so the reaction center is kind of in the middle here, right? And this is where we can finally transfer that electron to a molecule where it can be captured. All right? So if we look at our primary electron acceptor, all right, this is a molecule that's going to capture that electron that has been excited. All right, once it's been captured, we can transfer it along to other molecules instead of having it bounce between all of our chlorophyll pigments. Now the reaction centers are really what plays the major difference between these 2 photosystems. All right, so photosystem 2 has a reaction center that we call P680. All right, and P680 sometimes we'll put a little asterisk behind it. Right, that means it has an excited electron. All right, photosystem 1 has a reaction center that we call P700. And again, once it becomes excited, we have that little asterisk behind it. Right, so they're called P680 and P700 depending on very, very specific wavelengths that they pick up. All right, so once P680 and P700 has that excited electron in its reaction center, right, this is really, really unstable. Right, so the energy from that electron has to be transferred really, really quickly. Right, and this is where that electron is captured and is transferred to a primary electron acceptor. All right, it's just this generic square in our diagram here. All right, so that's the anatomy of these photosystems. Now they are a lot more complex than just this, all right, but we're just going to focus on the very basics. All right, so let's start working through this step by step. So the first step, all right, is going to be in photosystem 2. So in photosystem 2, right, light is coming in, right, and it's exciting an electron. Right, that electron is being excited, it's being captured in that reaction center, right? And eventually, that electron is going to move out of photosystem 2 via that molecule that captured it and be transported into the electron transport chain, which we'll talk about in just a bit here. All right, just again, we're capturing light energy. So we have our light coming in, it's hitting a pigment molecule. It is bouncing around from pigment molecule to pigment molecule, right, until it reaches the reaction center, where it's still relatively unstable, and then it has to be transferred to this electron acceptor, right? It has been captured when it's been transferred to its acceptor. All right, now one of the most important things, especially from a human standpoint here, is that photosystem 2 uses water. All right, so down here we see that H2O, water, is being split into oxygen and hydrogen. All right, as part of being split apart, those electrons are moving into the reaction center to replace the one that was lost because it got excited. All right, so essentially we're creating oxygen in this step. All right, so photosystem 2, we are breaking apart our water molecule. Right? The electrons are stripped away from the water molecule and go into our reaction center to replace the one that was lost. Right? As a byproduct, we get oxygen. We also get some hydrogens. Right? Now, eventually, these hydrogens will be useful in making ATP. Right? We're going to kind of ignore that part of it for the time being. Right? But know that this is where we are creating oxygen. All right, so we have our electron, right? It got excited in photosystem 2, it got captured in photosystem 2. Now it is leaving photosystem 2 and going into the electron transport chain. So the electron transport chain is actually a series of molecules that help carry that electron throughout this chain reaction here. All right, so it's a bunch of molecules, all right, and electrons being transferred between them all in order to get to photosystem 1. Right, now one of the biggest molecules, most important molecules in the electron transport chain is this cytochrome complex. Right, so this cytochrome complex, when it receives the electron, right, it moves hydrogen ions from the stroma into the inside of the thylakoid, into the thylakoid space. All right, so the movement of electrons through this complex moves these hydrogen ions into the thylakoids. And this, again, is going to become important when we talk about creating ATP, right? That energy currency of the cell. All right, so we've gone through the electron transport chain. The electron has finally made it to photosystem 1. All right, in photosystem 1, the primary goal is to create this molecule called NADPH. Right? It's an electron carrier. It helps carry these electrons to be used in other processes in the cell. You can kind of think of it as an energy currency. Right? Now, instead of P680, we have P700 here in our reaction center, but it's set up the same exact way as photosystem 2. Right? The only thing that is really different is that we're not splitting water at this photosystem. All right, so at this point, all right, we have our electron that's coming in. It has lost a lot of energy because it's been transferred to so many molecules. Right, so we have to re-excite this electron. We have to give it some more energy. Right, and again, we're using light to do this. Right, light is coming in. It's bouncing from pigment to pigment to pigment. Right, which is going to excite that electron and bring it to a higher energy state, so that it can go on to continue the light reactions. All right, so once our electron has been excited, all right, and we have this diagram here showing that the amount of energy has increased. It's a lot higher because it's been excited by light again. All right, it's going to be passed to multiple molecules before it reaches this molecule called ferredoxin, I'm not too worried that you know this molecule. I put it up here because it's that last molecule that the electron is at before it's used to create NADPH. And this is where that last molecule that we talked about comes into play. The enzyme, right, which helps speed up reactions, NADP plus reductase, comes into play. Right? So NADP plus reductase is going to aid the process of turning NADP plus, right, which is essentially missing hydrogen and missing some electrons. It's going to add that missing hydrogen, add those missing electrons back into the molecule in order to create NADPH. All right? So we're essentially recycling this molecule here back into NADPH. All right? NADPH is where that electron is going to finally end up. Right? The electron has completed its journey at this point. All right, so big overview picture of the light reactions. A lot of times it's talked about as the Z scheme, right? Because that electron is going through different states of energy. All right, so if we look at photosystem 1 here, we have light coming in, exciting that electron. We see that its potential energy here has increased quite a bit, right? It's going through the electron transport chain, the cytochrome complex, before reaching photosystem 2, right? It's lost a lot of energy along the way, right? So now it's going to be re-excited by light and reach an even higher energy state, right? Again, it's going to lose some energy on its way through different molecules before finally being captured by NADPH. All right, that's a lot of information. Do I have any questions so far on the light reactions? All right, so we have the light reactions. We have followed this electron through all these different molecules. And I mentioned that at some point, all right, we're going to create ATP. All right, so we're going to create ATP using ATP synthase, which is a large enzyme embedded within our membranes here. All right, and this enzyme is powered by a gradient of hydrogen molecules. All right, meaning we have a lot of hydrogen molecules inside of our thylakoid versus outside. Now these molecules are going to want to go from an area of high concentration to an area of low concentration, right? And it's going to move to this area of low concentration outside of our thylakoid membranes by going through the enzyme ATP synthase. All right, so we have these hydrogen ions moving through ATP synthase. As it moves through this molecule now has enough energy to create ATP. All right, so it's not creating ATP out of thin air. It has some inputs. All right, the inputs being ADP plus a phosphate group, combining it together to make ATP. All right, big picture here is that this gradient of hydrogen ions are causing enough energy as it moves through this ATP synthase, right, to combine molecules together in order to create ATP. All right, so I mentioned a couple of ways that this gradient was set up, right, where we have higher amounts of hydrogen ions inside the thylakoid versus outside. All right, one is by the splitting of water. Right, so if we're splitting water apart, we're releasing oxygen, but we're also releasing these hydrogen ions. All right, that's going into the thylakoid lumen, increasing the concentration there. We also have the cytochrome complex. All right, so as the electron moves through the cytochrome complex, it is pumping these hydrogen ions into the thylakoid space. All right, so that's another way that we are increasing the number of hydrogen ions inside of our thylakoids. All right, but we also have another way that's a little less intuitive to think about. All right, we're decreasing the number of hydrogen ions in the stroma, all right, so outside of our membrane, by adding it to NADP plus to create NADPH. So we're decreasing the amount even further on the outside of the stroma. Right, so we've set up this very nice gradient now in order for these hydrogen ions to move through and help create ATP. All right, big picture takeaway. We have created three main products from the light reactions. All right, we've created oxygen. We had to split apart that water in photosystem 2 in order to replace the lost electron. All right, so that gives us oxygen. We are creating NADPH, which you can think of as an energy currency. All right, so that's the very end product of the light reactions. We're also creating ATP. All right, ATP is a huge source of energy currency within our cells. All right, and we're creating lots of it by setting up that gradient, right, and using ATP synthase. All right, have I lost anybody yet? Sounds good. So that was a lot of me talking. I think this animation does a really good job of kind of showing you visually what's going on.
The thylakoid membranes contain specialized molecules that work together to perform the light reactions. Light is absorbed by protein pigment complexes called photosystems. There are two photosystems, photosystem 1 and photosystem 2. The photosystems transform light energy to chemical energy by exciting and then shuttling electrons from molecule to molecule in a chain-like fashion on the thylakoid membrane. This process is called an electron transport chain. Let's take a closer look to see how this process works. First, photons of light hit chlorophyll, a light-absorbing pigment in photosystem 2. Electrons in the chlorophyll are excited to a higher energy level. The excited electrons are passed to an electron carrier. Meanwhile, water splits and releases electrons. These electrons replace those lost at photosystem 2. The byproduct of this reaction is oxygen, which is eventually released into the air. The other products are protons, or hydrogen ions, which are released into the inside of the thylakoid, or lumen. The excited electrons move to the cytochrome complex. Some of the energy from the electrons is used by the cytochrome complex to transport additional protons into the lumen. The second electron carrier, a protein inside the lumen, receives the electrons and passes them to photosystem 1. These electrons have now lost most of the energy they gained from light in photosystem 2. Photons of light hit chlorophyll in photosystem 1 and excite the electrons again. The electrons are then passed to the third electron carrier. Finally, these electrons are either recycled or they interact with an enzyme and NADP+, the final electron acceptor of the light reactions, to form NADPH. Some of the energy from light is now stored in the reduced molecule NADPH. Some of the energy released from the transfer of electrons established a proton gradient across the thylakoid membrane. Protons that accumulated in the lumen diffuse into the stroma through an enzyme called ATP synthase. ATP synthase uses the potential energy of the proton gradient to combine ADP with inorganic phosphate to form ATP. In this way, the potential energy is transformed into chemical energy stored as ATP. ATP and NADPH now have stored energy from the light reactions. This energy can be used in the Calvin cycle. This light-driven electron transport chain is usually continuous in the presence of sunlight. It encompasses a series of chemical reactions that involve light absorption, energy conversion, and electron transfer carried out by the photosystems and other enzymes on the membrane of the thylakoids.
All right, so I think that video gives a great visual and kind of puts it all together for you here. All right, so I want us to, question.
Why does photosystem 1 come after photosystem 2?
Because photosystem 1 was named 1 because it was discovered first. Very confusing. So they discovered the second step before they discovered the first step. So that's why it comes after. Yes. I feel like sometimes they just named them that way just to confuse everybody that has to go through like Bio 101 or a basic science course. Any other questions? All right, so let's step back. Let's try to answer this question here. All right, if there was a mutation that caused photosystem 1, or that affected photosystem 1, I should change that wording here, would it affect the production of oxygen?
Initially, I wanted to think
We're getting a little lost over here.
Oh,
Oh, all right. Okay. I thought that was like a word like.
no, it wasn't like a bad.
or if it's just like the focus for one cell versus like the entire.
All right, so looks like we've maybe mostly answered the question here. All right, so if there is a mutation that caused photosystem 1 to stop working, all right, would that affect the production of oxygen? No, yeah. At first, it definitely wouldn't affect the production of oxygen. Something that I probably wouldn't expect you to think of is that eventually that whole system would kind of get blocked up, and then we might see a decrease in oxygen, right? But definitely not at first, right? We don't split water in photosystem 1. That is photosystem 2. All right. So let's talk about the second half of photosynthesis. And I actually think the second half is a lot easier to think about. All right, so the second half would be the Kelvin cycle, often called the light-independent reactions or the dark reactions. All right, they are using ATP, NADPH, and carbon dioxide to create sugars. All right, now notice that the light reactions, we created NADPH and ATP. We are now using those molecules that we created in the Kelvin cycle along with carbon dioxide in order to produce sugars. All right, so this slide is more a big picture concept that we're going to go through step by step here.
Sorry to like backtrack a little bit.
Oh no.
But so like when we watched the video about light independent versus dependent,
Yeah,
this one is independent, right?
independent. Yes,
Okay.
yes. We're not actually using light in this process.
And that's why it's called like light independent.
Yes.
And then the other process is light dependent.
Yes, because that's where the photons are actually coming in and exciting those molecules.
Okay.
Now they rely on each other, right? So the light reactions creates ATP and NADPH that the Kelvin cycle relies on, right? But the Kelvin cycle could still work at nighttime, right? And some plants will actually use the Kelvin cycle at night rather than the day, just as an adaptation to maybe heat stress. All right, so big picture about the Kelvin cycle, right? We're taking in CO2 from the air and incorporating it into a carbohydrate, right? So this is we can think of it as a type of sugar, right? And the carbohydrate that is created is glyceraldehyde 3-phosphate, all right, abbreviated as G3P. All right, now G3P is the end product of the Kelvin cycle, but will go on to create glucose in a separate series of reactions. Right, now this whole cycle takes a massive amount of energy, right, which is why we have our light reactions to create the energy, because it takes a lot of ATP and a lot of NADPH, right? So for every 6 molecules of CO2, right, it takes 18 ATPs and 12 NADPHs. Right, so that's a lot of energy that this cell is needing. All right, so this is the Kelvin cycle in its entirety. And don't worry, I am not going to make you memorize every single molecule along its pathway. All right, we're just going to stick to the big important ones here. All right, but if you notice, we kind of have these three different colors in the background that splits the Kelvin cycle into three different phases. All right, so the first phase, in kind of this tan color up here, is carbon fixation. All right, we are taking that carbon from carbon dioxide and fixing it into another molecule. All right, so we're taking CO2 and putting it together with another molecule to create a separate chemical compound. Now the next phase, right, is called reduction and carbohydrate production, right, because essentially we're taking that CO2, right, we're incorporating it into the molecule, we've created an intermediate that is now going to go through a series of reactions to create more intermediate molecules before finally becoming a G3P, right? So that G3P is our carbohydrate that we have produced that will go on to create glucose as well as some other sugars as needed. All right, our last phase, all right, we have to regenerate a molecule that was initially used up, all right, and that molecule is RUBP. So initially we used it up, and in this phase we are essentially recycling it back into RUBP in order to be used again. All right, so if we zoom in on that first phase, carbon fixation. All right, so in carbon fixation, right, we're taking CO2 and we're combining it with this molecule called RUBP. All right, once we've combined it with this molecule, it's become a different molecule that I don't expect you to memorize, all right, but a molecule that's abbreviated as 3PG. All right, so we're fixing it initially to this molecule RUBP. You can notice I have bolded this molecule in our slides. So this is one of those few molecules that you might want to know the name of.
Which one?
RUBP. So it's this molecule right here that I'm circling. All right, so that's the initial molecule that CO2 is going to combine with. Now it combines with this using an enzyme called Rubisco. All right, so an enzyme is just going to help speed up the process of combining the CO2 with RUBP. All right, Rubisco is a very important enzyme that we'll talk a little bit more on later. All right, so essentially we have created a product, all right, that is a 6-carbon intermediate. Once we've combined it here, we have 6 carbons in our molecule. All right, and it's going to be split into 2 to create 2 different 3-carbon intermediate molecules. All right, so those 3-carbon intermediate molecules are going to go on to our next step, all right, phase 2, all right, and this is where we start seeing the input of energy. All right, we can see ATP is used here in order to create another separate molecule from 3PG, all right, creating yet another molecule. We see NADPH is needed to create that second intermediate into yet another molecule. All right, so we're using energy to create different molecules in our cycle, and as a result, we're going to create the molecule glyceraldehyde 3-phosphate, or G3P. All right, so that's the molecule that we want. All right, that is the molecule that's kind of the end product of our Kelvin cycle. Now this G3P will eventually go on to make glucose, right? But if you notice in our cycle here, we've incorporated 6 carbon dioxide molecules, and as a result, we now have 12 G3Ps, right? So G3Ps, that's the molecule we want. We got 12 of them, but not all of them are going to go towards glucose, all right? Only about 2 of those 12 are going to go on to create sugars. The rest of those molecules are going to continue in on our cycle here. All right, so we have our last phase, where we're taking those 10 G3P molecules and we are using them to regenerate RUBP. All right, so remember RUBP was initially used up in the first phase, right? It combined with CO2, it was used up, and then it went through all these different transformations to become 12G3P. Well, now we got to get that RUBP molecule back, all right? And this is that last phase, all right? We are taking our 10 G3Ps and converting it back into RUBP, right? Using quite a bit of energy, quite a bit of ATP along the way. All right, so once we've converted it back to RUBP, this cycle essentially starts over again with another molecule of CO2 being fixed. All right, so it's very much a cycle. The molecules being used up, transformed into sugars. Some of those sugars will be made into glucose. Some will go on to regenerate that initial molecule, which can again go through that cycle. All right, any questions about that cycle in particular? All right, like I said, I think this one's a little easier to think about than all those different molecules and the light reactions. All right, now I mentioned Rubisco being that enzyme that helps incorporate CO2 into RUBP. All right, Rubisco is a really, really important enzyme, all right, and it's thought to be the most abundant in the world. All right, most abundant enzyme on Earth. Every plant that goes through photosynthesis has this molecule, right? It's super important. Without it, gosh, I don't even know. It would be much less efficient, right? Rubisco, super extremely important. All right, so I put this activity up as homework that you can do on your own, just to kind of draw it out, get that muscle memory, make sure you understand what is going on. All right, so in your drawing, and again, you don't have to be an artist to do this, I just want you to include all these different molecules, right, and show me how the light reactions are happening through your drawings. All right, now I put it as an out-of-class activity because it might take a little bit of time because there are a lot of molecules and are a lot of labels to incorporate. All right. Any questions about photosynthesis? I threw a lot at you. I will be here to answer more questions about photosynthesis tomorrow once you've had a chance to maybe digest the information a little bit. All right, so for homework tomorrow, all right, I'm going to introduce this right away. There's an assignment on a Gardeners of the Galaxy podcast. All right, it's an easy one. Listen to the podcast, answer the questions that I have posted on Moodle. Make sure you do your drawings and turn them in to me tomorrow. And then you have a reading and a video like normal that you will take the quiz on. All right, and just as an upcoming, all right, exam 1 on Monday and endurance on Tuesday. All right, so we have not taken the reading quiz for today yet. So let me make sure I have the right password for you here. All right, we'll go ahead, we'll take the quiz, and then the last thing we do is I kind of have a movie day planned, all right? I have this really cool documentary called How to Grow a Planet, which I think fits in very well with our topic.